Great pleasure for me to introduce today's speaker, Kafui Ziraza from Duke. Um, I think it's fair to say that you have a very long and close association with Duke. After graduating from UMBC as a Mayhoff Scholar, Kafui went to Duke as a graduate student in Miguel Nicolelli's lab, and then started, I think, a short postdoc, relatively short postdoc, also in his lab, and then decided to complete his training by going into medicine, and did his training, medical training, also at Duke, and his residency, and then became an assistant and then associate professor still at Duke. Kafui, from his early interest in neuroscience, really had three passions, I think, that closely interacted with each other. One is a principle of engineering, and another one is neuroscience, and the third one is mental illness. And I think that throughout his career, both as a graduate student, as a postdoc, and then as a faculty, he's really been very eager to tackle some of the most complex issue of complex neuronal dynamic, try to decode them and harness this knowledge to try to understand mental illness, but with always a goal and try to at least think about how this knowledge and these advances can then possibly help human patients. I had the great privilege of interacting with Kafui, who was a member of the Brain 2.0 working group, trying to think, review the Brain Initiative, and think about new development of the Brain Initiative. And Kafui was an extremely engaged member, and I take advantage of this platform to thank him, and also for his really visionary ideas also on how to use basic science to help patients, but also how to reach out to the largest public and to make sure that advances in neuroscience will benefit not only few privileged folks, but really the large public in general. And I must say that his passion for science equaled his passion for outreach and a number of issues in science and science policy. Kafui has received quite a number of awards, including in 2016 the Presidential Early Career Award. 2016, that was the right year, right? I'm glad you didn't have the year after. And on this, it's really a great pleasure, and help me welcome Kafui here.
Thanks so much, Catherine, and thank you all for having me here. As Catherine mentioned, I've been at Duke for almost 20 years, both as a medical student, a graduate student, started my research lab, and then started psychiatry residency soon afterwards. And so I'm a bit of a glutton for punishment, and the real challenge for me has been to take some of these thoughts I had originally being trained as an engineer, fusing that with neuroscience and psychiatry, both to come up with new treatments for the devastating disorders that we see, but also how to use that knowledge to understand how information is organized in the brain. And so there's a real question that I've been wrestling with, and some of my thinking has evolved around this question over the last year, but it's this question of what causality is, right? And when I use the word causality, it's this question of how does the brain do what it does? And I spent a lot of time thinking about this question, and I was having a fascinating conversation with one of my colleagues who's a statistician several years ago. And I was telling about all these great neuroscience tools that we have. We have optogenetics, we have DREADS, we have the ability to start to stitch these things together. And the promise that this has, both to tell us how the brain works, but then ultimately to translate these things into new treatments. And my colleague asked me a fascinating question that I've literally been wrestling with for 2 years, and I haven't quite solved yet. And some of this actually came up as we were putting together Brain 2.0. And the question they asked me was, so Kaf, right? Surely you believe that the reason it's like hotter in the summertime and colder in the winter, and the reason we have seasons and it's like warmer during the day is because the sun, or the earth is going around the sun. And I said, of course, like what a stupid question to ask, right? And my colleague paused for a minute and they said, yes, but you've never manipulated the earth or the sun, right? So why is it that you can conclude that there's causality without the need for an intervening manipulation? And I wrestled with this question for a while, and ultimately what I concluded was that the reason I believe that is because people have observed the system over and over and over again across time, and they've come up with underlying principles of the system, things like gravity and radiation, and pooling those together, they're able to come up with a model of the system that we can then test in subtle ways, like putting up an umbrella and realizing that it's cooler in the shade than it is outside of the shade. And there's certainly other hypotheses that one could have for why it's hotter in the summertime. One of them might be, if you look around, there are more people outside in the summertime. The human body is 98 degrees Fahrenheit, so perhaps the reason why it's hotter in the summertime is because human beings are outside making it hotter. And there are lots of ways to violate that assumption as well. If you go to parts in the world where it's really hot, no one's outside in the summertime, like, you know, whether it's Texas or somewhere in Saudi Arabia, right? And so you can observe the system and then violate your understanding of the system. And what I'm going to do is I'm going to spend almost the first third of the talk talking about this idea of a conceptual and theoretical framework, and then I'm going to build in the engineering and the neuroscience on top of that theoretical framework to get at this idea of causality. How do things work? And one of the key points that I wrestled with some of my colleagues over the last few months is this idea of the challenges that come along with correlations, right? So correlations are not causality, right? So things can be correlated and they are not the cause of it. But what is important to keep in mind is the absence of a correlation is the absence of causality. And so what you can do is observe a system repeatedly, see where the correlations are not present, and thereby reduce it to the dimensions of the system that might be causal. And I'll talk a lot about the strategy or part of a field called machine learning, where the idea is to look for the absence of correlations so that you can see what's remaining and build a model system around that. So that's a framework I'm going to use around this talk, this idea of these challenges of getting around how the brain does what it does to produce behavior. All right, I'm going to frame this talk around depression. I choose depression because it is the most debilitating mental disorder in the world, according to the World Health Organization. And with disability, this is years of productivity loss. So people think about things like Alzheimer's and Parkinson's disease. This is the most debilitating disorder in the world, defined according to 9 behavioral criteria. I won't spend a whole lot of time here, but depressed mood, diminished interest, gaining increases or decreases in appetite, sleeping too much or sleeping too little, moving too much or moving too little, fatigue or loss of energy, worthlessness, guilt, decreased ability to think or concentrate, and recurrent thoughts of death. You have to have one of the first 2 and a combination of 5 out of these 9. And as I started residency training, it was pretty clear if you start observing patients that folks can have the same diagnosis of depression and no overlapping symptoms whatsoever. In fact, there are about 1,497 possible ways of getting depression. And that might be why our medications aren't so effective. So I trained at Duke, and I wrestled with how to think my way through this idea of depression and depression treatment, and came across this strategy that was one taken advantage of by our cardiology colleagues. And the idea here is that you can have the disease state, and I'll use that word a lot, disease state, and I'll paint that as the talk goes along. But you can have this disease state, in this case we'll call it heart failure. So your heart is an organ, it pumps out blood, and the disease state is physiologically it cannot pump out as much blood. You call that heart attack or heart failure. And there are medications that we can use, things like pacemakers or things that make the heart contract a little bit more, that take the diseased heart and make it pump more blood out. So this idea of taking a diseased heart in red, make it function more normally in green. And that's what the medications do. And about 60 years ago, cardiologists took a slightly different approach, or epidemiologists. And what they wanted to do in that case was take a young population and track them across time. And they say that we know a constant risk factor for having heart disease. In that case, it's aging. And so can we, instead of looking at folks that have disease and make a diseased heart function better, can we actually segregate individuals out before they have heart disease and figure out if there are new biological pathways that one could target? And when they did that, this is a Framingham study, what they found was there are things that you could target, like blood pressure and cholesterol and smoking. So these are biological pathways that one can target to prevent a heart that would otherwise be diseased from ultimately moving in the disease state. Rather, you can have it move along this trajectory with the constant risk factor of aging. And I started to wonder if you could take the same sort of strategy with something like depression. And so I'm going to orient you to the terminology I'm going to use throughout the talk. The disease state here, major depressive disorder, I'm going to use the terminology susceptibility This will allow you to orient it to some of the other literature, in the field. The idea here is if you have the disease state or susceptibility, antidepressants or the treatments make your brain function more normally, right? And we'll call that state resilience, right? So susceptibility is the disease state, resilience is the functional state. Instead of aging, and here we'll call it severe psychological stress, I observed repeatedly in the clinic that stress made these illnesses show up, stress made people worse, stress made people relapse if they had been appropriately treated. And what we're going to try to do is segregate these individuals out before the disease state shows up. And the terminology we're going to use, instead of blood pressure, cholesterol, we're going to call this vulnerability. So is there a latent state that exists in the brain called vulnerability that we can identify that says individuals that otherwise we'd be unable to distinguish them, individuals with this latent state of vulnerability in their brain, if they are stressed, they will move this way. And we're going to try to see if this state exists. So this is our objective, right? To our wonderful colleagues in other areas of neuroscience who study sensory systems or motor systems, we have a profound problem in doing this in animals, which is, right, the things that bring animals in the clinic, we do a really poor job of measuring without a self-report, right? So you can't ask an animal if it's guilty. You can't ask an animal, besides a human, you can't ask a non-human animal if it's guilty or suicidal. And if you do, it generally doesn't answer. And we were struggling for a very long time with how to make this problem a little bit more tractable. And we thought one sophisticated approach we could do is borrow some recent approaches that were used by our colleagues in the human imaging side. So I'll do a quick orientation to this slide. It's a human fMRI study, functional magnetic resonance imaging. They're basically looking at the whole brain and looking at changes in blood flow to different parts of the brain as a proxy of cells being activated. And so they're looking at whole brain activity. And what they're doing in this task is they're bringing in a bunch of healthy controls. I don't like calling them normal subjects because they're Duke students, but they're bringing in a bunch of healthy controls and putting them in a scanner. And then they're showing them videos, right? And these videos are basically they're high emotional content. So these are videos that are happy or sad or angry. They're showing videos while they're looking at the whole brain. And then they're taking this activity that they get out of the scanner and they're saying, is there a pattern of activity in the brain that matches the type of video that we're showing them? So there's a pattern for happiness, is there a pattern for sadness? Can we essentially decode the whole brain and match the pattern with the emotional state using a set of tools called machine learning, which I'll come back to. And then they send the kids home, bring them back the next day, put them in the scanner again, and say, don't think about anything, which smart people are really bad at. And after 20 minutes, they tap them and they say, well, what were you thinking and feeling? They get a self-report, then they say, how well does what your brain scan today said you were feeling match what you were feeling? And they're able to do this relatively well. They can match, they can classify what your brain looked like against what you reported that you were feeling. And what this means is that now you can extract an emotional state out of a brain without the need of a self-report. And perhaps this is the type of strategy that we can employ in our animal models. Induce states, decode those states, and then see if those states show up again when we're not inducing them. All right? As I go through the talk, I'm going to frame assumptions that I pull out of each of these observations. Some of these observations, they won't necessarily be what you see on the screen from the data before, but I'm going to show you as the thinking evolves. So the first one, which is an interesting observation, is fMRI, the time scale of fMRI is on the order of seconds, right? So you're getting pictures of the brain on the time scale of seconds. And one of the things I think is really important about this observation is that if you're thinking about setting up a system to do vision or motor function, you want the system to resolve information and generate information really quickly, right? Or you don't get dexterity unless your motor system can generate millisecond timed information. But I'm not sure it makes entire sense to generate a system that's generating happiness or sadness that's resolved on the order of milliseconds, right? I never had a patient complaining about being sad for a quarter of a millisecond, right? And so perhaps what the brain does when it's generating these states, it generates latent states that have time scales where they evolve, manifest, and stay present over time scales that are longer than milliseconds. Perhaps it's seconds. So the assumption that we're going to pull out, and you'll see how this comes in the model we develop, is that emotions are encoded at least at the seconds time scale. All right? The second challenge that we face is thinking about how systems are organized in biology, right? So if I was to do a cool set of experiments, right, I put up a bunch of enzymes and substrates. When I do this, I always say every medical student and every graduate student in the room knows exactly where I'm going with this. But if I was to say which of these enzymes and substrates contribute to how much that model organism is going to move, you might increase one and decrease one, measure how much the model organism moves as you're going through in your Excel spreadsheet, and marking down which of these contribute to how much the organism moves. And what you would ultimately conclude is that all of them contribute to how much the organism moves because they're not independent, right? So they're all part of the citric acid cycle. And the point of this system is to basically generate energy. And so these enzymes and substrates work together to generate the output. The output in this case is orthogonal to the system. And so it generates this emergent property, which is energy, by all of these parts of the system working together. And we're going to try to frame, as I move through the talk, to frame how a system of parts in the brain work together to generate an emergent property, and we'll call that emergent property emotions. Another example of an emergent property, if you think about this case, I'm really terrible at drawing, so this is a neuron. If you think about a neuron firing, a single neuron in this case, it's hard to think about what the emergent property here would be. But if I took this single neuron and hooked it up to 4 other neurons, the emergent property becomes pretty clear. It would be a seizure, right? Because the system would activate itself over and over and over again. And if I change those neurons for metal, right, and I turned it into a metal ring and send electricity around, again, if you're from a physics background, you would understand what the emergent property here is. It would be a magnetic field, right? And so the system by itself, each element doesn't generate the emergent property. It's all of the parts of the system working together. So the second assumption of the talk is that emotions are going to be an emergent property of lots of brain cells working together. And the example I give of this is think about something like sleep, right? There might not be a single cell in the brain or the cortex or another area of the brain that is sleep. Sleep is a state that the whole system enters into because of the way lots of the elements of the system work together, right? So what we do in my lab is we record lots of the parts of the system simultaneously. This is a mouse. We can simultaneously implant electrodes into multiple sites of the brain. This is a CT image that we've co-rendered onto an fMRI. This is actually a rat brain. We do work primarily in mice though. And we can record many sites of the brain simultaneously. We're able to get local field potentials, and I'll talk about what those are on the next slide, as well as single units for multiple brain areas. This is 16 simultaneously in a mouse. It recovers just fine, and we're able to extract electrical information from many sites in the brain simultaneously in freely behaving mice. The local field potentials, this is an example of what we see. If you're on the human side, just think EEG, so scalp activity. We're getting these from the tips of electrodes implanted in the brain, so we can localize them much better than what you get when you're recording from the scalp. These local field potentials, we can filter them in different frequencies and quantify how much activity there is in different frequencies. So each brain area, we can resolve in frequencies and see how much activity or power there is in each of those frequencies. We can also take advantage of engineering principles, and those engineering principles say things that change together over time tend to fall within the same system. So we can look at those local field potentials and see if they change together over time, and we can quantify how much they change together over time. We can do cool mathematical tricks offline. We can shift one relative to another and ask if they get better coupled. In other words, can you predict information in one area in the future because of current information in the other area in the present? And from that, we can infer the way information is moving. It's the same type of thing that's done with statistical forecasting, right? You can infer that information is moving from area A to area B because future area in B, information in B looks like current area in A. We can also shift in the other direction and ask if information is moving in the other area. All right. Now, the last thing that is really important is I make no claims whatsoever what these local field potentials mean, right? I'm not even claiming that they are important information that the brain uses to generate behavior. What I will suggest, which I think is fair for what I'll point out today, is that these things reflect the activity of cells, which we will all argue are relevant for brain function. So if you look at the local field potential here, I'm just showing you that the spike timing of a local field of a neuron recorded from the same wires of local field potential is firing at a phase of the oscillation of the local field potential, and this is quantified as phase locking. And here I'm just showing you another property that we're able to decode in which a cell firing in prefrontal cortex is encoding the relative phase of the oscillation between two different areas, but not each one in isolation. In other words, that cell firing is encoding the synchrony between two areas. The first one is encoding the phase. We can also find cells that encode the amplitude of oscillations. So these local field potentials, which are thought to reflect populations of neurons, right, are capturing lots of neurons firing together. So in other words, they're capturing the emergent state or the emergent properties that come out of neurons firing together. So at a minimum, these local field potentials reflect the activity of lots of neurons. And we'll leave that as our last assumption, right? So the local field potentials reflect the activity of populations of neurons. Think an emergent feature, right? So we can capture whatever those dynamics are. So several years ago, we got to the point now where we're recording local field potentials across multiple brain areas. We're trying to figure out how to map these onto behavioral states. and you can immediately appreciate the data challenge that you get into, right? You're recording local field potentials, which are resolved across frequencies. They can synchronize across brain areas. And the way folks would typically solve this challenge is to record hundreds of thousands of mice, right? That's how you deal with the statistical problem. But we clearly were not implanting hundreds of thousands of mice to figure out what depression or stress states look like in an animal's brain. So Duke had launched an information initiative, and it was this whole center around making data and models around data more accessible to the community at Duke. And so I set out a meeting to meet with the head of the information initiative at Duke, a guy named Rob Calderbank. He spent most of his time looking at telecommunications. And he would go on like the next year to win the Shannon Prize, which is like the data science equivalent of like the Nobel Prize. And so I'm describing to them, gave him like basically the same background that I just gave you about the brain and causality and all these things we're measuring in depression. And I finished up, about half an hour took me to explain all this to him. And he says, that's really cool. It sounds a lot like geothermal imaging. Now, my immediate response was I had done something totally incorrect. So I explained to him neuroscience all over again from scratch and our assumptions. He said, no, geothermal imaging, imagine it. You have a planet, right? You're trying to study this one planet. You've got satellites going around. You're integrating information from multiple sources to figure out what's going on with this one thing, to zoom in and figure out what people are growing in different parts of the world based on this integrated information. And so when he described it that way, we went back and forth. He introduced me to somebody else, and he says, I really want you to think about how to create models that might exist in other parts of the engineering space and think about how those might map onto the brain. Before you knew it, I found myself on multiple thesis committees for electrical engineering students. I'm an engineer by background. And I will say this was probably one of the most important points that I picked up. I very quickly found myself on thesis committees reading prelim documents where, like, it's not that I didn't understand what they were writing. I'd never seen symbols in half of the formulas on the page, right? Like, there was no, like, interpreting this, right? I just never had seen the symbols in my life. And it took almost 2 years to figure out how to bridge together the space between what I was seeing in electrical engineering onto the neuroscience to start to solve some of these problems. But I'll show you the model that we came up with. So the model looks like this. It's hierarchical, so it's got multiple layers to it. And we decided that these layers would each belong to some of the assumptions that we'd made up to this point. So they decided that the bottom layer would be mine, right? The bottom layer would be things that I could record in the brain on that milliseconds timescale. So these local field potentials with their features, and it would be the observations. And these observations would be all the local field potentials from each of the brain areas. They would allow those observations to be resolved across frequencies. So we can take all the brain areas, we could take all of the frequencies that were reflected in the oscillations for each of those brain areas, and all of the possible combinations of how all of these oscillations could synchronize with each other. So basically 5,000 possible observations that we're getting per mouse, and we can observe those changing across time. And then, so that's layer one, milliseconds timescale. Layer two was going to be based on that seconds timescale that we'd seen in the fMRI data. And the way I would think about this is if you had an orchestra, you have individual instruments. The instruments can play sound, which is pitch, which is just oscillations that are really fast, right? And somebody's got to take those instruments and coordinate them together. So that's this layer, right? There's some sort of conductor that is causing the instruments, which are making really fast sounds, to play together at a slower time scale, right? So we're going to integrate milliseconds into a seconds time scale together. So that's a time integration that's happening. And that's going to be organized into multiple networks. So network one might be observation 1 and 7. Network 2 might be a little bit of observation 4 and oscillation 8. But it's basically going to organize the fast things together into slower latent states or networks. And then the highest level of the model, we'll call that task labels. If this was our Duke students, this would be happiness or sadness or anger. So sadness might be a sprinkle in network 1 and 8, sadness might be 4 and 12, right? And what the model is going to do is it's going to learn what the red lines are. So it's going to learn what the networks are made up of and what the emotional states are. And obviously, we're working with mice, so we don't get to call them happy or sad. We don't get to put them in an fMRI scanner, but we're going to develop a task that basically mirrors basically what they tried to do in the fMRI state. All right. And it looks like this. We're going to start off, and we're going to record our mice in 3 settings. So they're implanted with electrodes. First, we're going to record them just hanging out in their home cage. This is our resting state. Mice are doing what they do, like eat poop when they're left alone, just sort of relaxed. Then we're going to take the mouse and we're going to put it in an empty upside down cup. Think of this as like not super restrained, almost like jail, a little bit stressful, but like, okay, the animal's relatively safe. And then we're going to put a big, nasty, aggressive mouse. It's about 50% bigger and angry on the outside. So this is a little more like the Shark Tank, right? A higher threat state that the animal's in. And what we're doing in this case is we're not interested in if the animal's running around, right? We've sort of limited its behavior repertoire. We're interested in how its brain changes in response to each of these stimuli, right? Okay. And then the way we're going to get into a depression model is we're going to take advantage of chronic social defeat stress. This has been used in multiple labs in the literature. And what they do is they take a C57 mouse, get these mice from Jackson, and we're going to take it and we're going to take this CD1 mouse, which I mentioned is about 50% bigger. We're going to get the retired male breeders, which makes them a little bit more aggressive. And then when 100 come in the lab, we're going to take the 10 most aggressive. And so these are really big, nasty, aggressive mice. And we're going to let them hang out in their own environment, which also makes them protective. Then we're going to take our subject mouse and put it in the cage with the big aggressive mouse. This does not go well for the subject mouse, so we've got to end the encounter after about 5 minutes. And then you have to then it lives next to the animal that just beat it up. So 24 hours, it's basically there's a screen door in which it can hear and smell the animal that just beat it up. After that 24 hours, we put our subject mouse in with a new cage. It gets beat up by that mouse, lives next to that mouse, and we repeat this 10 days in which our mouse is getting beat up by 10 different mice that it then has to live with. I describe this as my childhood with my brothers. All right, so at the end of this process, the mouse has decreased interest in sugar and sex, anxiety-like symptoms, hyperactivity of its HPA axis, disrupted circadian rhythms, metabolic syndrome, and profound social avoidance. This has been used as a model of depression because the mice respond to treatments with multiple antidepressant agents if you treat them chronically, also acute ketamine. I think it's fair enough to just call this a stress-induced behavioral syndrome. I don't think there's a great argument for why this is PTSD or depression or anything else. It's just a stress-induced behavioral syndrome. But what's particularly important about this syndrome is it only happens to about 60% of the C57 mice. About 40% of them don't show any of this at all, right? So here's our experimental setup. We're going to implant animals with electrodes. We're going to record local field potentials in single units, and we're going to put it through our 3-part brain state assay, right? We're going to induce these brain states, and we're recording neural activity. Then we're going to take our animals, we've got about 50 of them, we're going to split them in half. Half of them then go through this paradigm where they get beat up over and over and over again. Half of them go back down the vivarium and they get like 10 days off just to be on vacation, right? And then at the end of that 10 days of getting beat up, we figure out which of the animals are susceptible, in other words, which ones didn't handle the stress well, and which of the animals are resilient. And then we look at everybody's brain all over again. So we look at their brains before, we look at their brains afterwards, and then all of a sudden, now we have those groups that I described in the cardiology study, right? We have everybody's brains beforehand, and then we have brains afterhand, some of which tolerated the stress well, and some of the brains that didn't. And what we're gonna ask and try to figure out is, are these brains in these C57 mice different before they were ever stressed in a way that predicts who would tolerate the stress well and who wouldn't? So instead of learning happiness or sadness, what we're going to get our machine learning algorithm to learn is what is the difference between a brain that's been stressed and a brain that hasn't been stressed? What is the difference between a susceptible brain and a resilient brain? And then what's the difference between a brain when it's in the first part, the home cage, the second part, like almost a jail situation, and the third part, which is the Shark Tank? And we'll learn that by learning the second state versus the other two and the third state versus the other two. So we're basically setting up our machine learning algorithm to learn these 4 things. Yes? Yeah.
Questions about the model set up here. So one was, the first was, when you drew the network, so you had arrows going from the electome to the task
Yeah.
label, right?
Yep.
Which seems a little bit strange to me because presumably the task is what you, the experiment, control, and that's inducing changes.
Yep. Yep.
Why don't the arrows point to the other?
Yeah, so in what we're trying to infer that there are emotions that show up in it. So even though we're inducing them, the networks are generating emotional states.
But the networks are not. Oh.
Right.
The networks are not. Okay,
So
it's just. I think it's a semantic thing that we call task labels.
Sure. Yes. Yes.
Okay.
Yeah.
And then the second question was, since you have this model, why do you need to train an SVM? Why can't you just compute the conditional distribution of task labels, conditional and electron factors? Why do you need to have a whole other discriminative model?
Because we're essentially going to learn. So what we're trying to figure out is which combination of networks together are encoding these, right? So sorry, let me make sure I'm understanding your question.
I'm just saying, if I understood that, so if you go back to the slide that you were just showing with the SVM,
Yeah, so right. So the SVM is actually, so what we're doing is we're essentially learning, and we're both learning which are the features that change together across time in the network, right? And that describes, that's basically the unsupervised. And then the task labels are basically pushing and tweaking the network so that they are also figuring out which combination encode the states. So the SVM is built into the training of the model.
Oh, I see.
Yeah.
Maybe I can add
Yeah,
more.
yeah. All right. So we basically learn these 4 networks, and I'll show you what these networks. So we're learning all the states. 4 of them are doing the decoding in this case, and I'll show you what the networks look like. And I'll warn you in advance, it's a super complicated slide, but I'll orient you to it. So essentially, along the diagonal, we're looking at power spectral densities. So it's telling you across frequencies how do the areas contribute to the networks, right? So this is suggesting that infralimbic cortex has a peak in the theta range or around the 8 hertz range that contributes to the network. And so this is what's going on in each of the brain areas. In the off diagonals, we have how the areas are synchronized together. In other words, these areas change together over time as part of the network. And so this is just showing you infralimbic cortex and BLA synchronized at this frequency, and that's part of the network. And so here's our visualization of that. We have our brain areas around the rim, frequencies from 1 to 50 hertz, and the tire part of the wheel says what's going on in each of those brain areas. So if you see color there, it means that this brain area in that frequency contributes to the network, right? And the spokes of the wheel suggest that these areas synchronize and they're part of the network, right? So the network is when these things literally show up on and off together over seconds of time in the brain. So these features show up together on and off repeatedly. All right. The other thing that's particularly important about this, and I'll come back to this throughout the talk, is when we're learning the networks, we also know how well represented they are across seconds of time, right? So you can have a network, imagine you have a bunch of instruments that are playing together, but they can play together, it's like a chord, right? The chord can show up on and off together over time. So we also get information about how strong each of the networks we learn are represented over time. And with each of the networks, remember I said the synchrony and the directionality of the information flow. We also have a lot of data about how information is flowing through the network in different frequencies. So it's a massive amount of data that we're pulling out for each of these. And I'm going to collapse it.
Yeah. So in the connection diagram there, it's just the amplitude, you're drawing the phase there?
Yeah. Yes, yep. And I'll get more into the details of this network and why we're like convinced ourselves they're not garbage, which I didn't believe at first. All right, so this is 4 of the networks, right? And I'm showing you the 3 groups of animals. This is all the post-stress data in those 3 states: home cage, empty, and CD1. And I split them into 3 groups: the non-stressed, the resilient, and the susceptible, right? And you can see that there are things that sort of naturally fall out here. We have a network. It's no difference between the susceptibles, the resilience, and the non-stress that basically looks like it's responding to the level of threat, right? As you go from home cage to empty upside down cup to the high threat state, in all 3 groups, the network basically is more represented. The thick lines is the average across the groups. Each individual animal is in the background. You have this network 2, which is just much higher in the animals that are showing the disease state or the depression state, right? So again, these animals aren't like running around. They're literally in a cup, right, or doing nothing. But there's a difference in the manifestation of this network strength in their brain. And then some other networks that are doing what's called multivariate coding. In other words, they don't separate on their own, but they separate in conjunction with other networks. The real reason we did this is because we're actually interested if these brains were different before they were stressed, right? So here's what the pre-stress data looks like. And we basically find 2 conditions in which the susceptibles and the resilience are different before stress. Again, the susceptibles and the resilience before stress are the non-stressed animals. So nobody is stressed at this point in time. So really pay attention to the difference between the green and the blue. One state here in which the susceptible animals just have a higher response of this network to the threat condition in their brains. These, again, are C57 animals. We're getting from Jackson. They all show up together. But the animals that have this higher response to the threat condition in their brains are the ones that are susceptible to future stress. So this is what we would call vulnerability. And this network 2 is also higher in the animals that will be susceptible in the future. So we basically have 2 candidates. These are just signatures. I'm not making any arguments about what these mean biology-wise. These are just signatures in the animal's head that is different in the animals that will be susceptible and the animals that will be resilient. One in this network and one in this network. So this is the point in time where I started asking, like, is this thing even real? Like, I've never seen any madness that looks like this. You data scientists are like way too complicated. And I need to convince myself that I need to look at something this complicated to segregate these animals out, right? Like we biologists like simplicity. And then secondly, that this has anything to do with like something a neurobiologist would believe, right? So, I mean, we have different areas, different frequencies, they're integrating, they're synchronizing, they have this weird directionality pattern of information flow from the areas. And the first thing we wanted to ask was just really simple. Couldn't you take one part of this network and equally segregate out whether an animal is going to be susceptible or resilient, right? So we basically take this thing and we chunk it down into pieces, right? BLA activity in this area, maybe synchrony in this area, and we ask if we just looked at that, could we predict whether an animal was susceptible or resilient? And so there's a measure here, the area under the curve of the receiver operating characteristic. 0 or 1 basically means you can separate out the two groups. These are the pre-stress groups. 0.5 means there's no difference whatsoever. And so we're just taking parts of the network, right? BLA, CEA by central amygdala on its own, ventral hippocampus, VTA. And then finally, we take the whole network. And we're seeing how well could we segregate out these animals just looking at an area on its own or parts of the network. And by far, the best classification is using the whole network. There seems to be somewhat of a signal in VTA that I'll come back to before the end of the talk, but it's about an order of magnitude different between what you can do with VTA alone and the network. This is on an AUC scale, so even though the numbers look close, there's a big difference between like 0.6 and 0.9, all right? So the first thing is you need the whole network. The second question is, remember I said these oscillations should be capturing cell firing in the brain or real dynamics in cellular activation. So we just asked the question, we took the network. This is the network activity resolved over time in red. And I'm just layering it on top of a cell that's firing in prefrontal cortex. And you can see that the dynamics of the firing of this cell are correlated with the network. Right? So we basically take all the 600 or 700 cells we have in the brain and we correct for multiple comparisons and see what percentage of those cells that we're recording show dynamics that match with any of our networks. And you can see for each of the networks, it's about 10 to 14% of the cells that we record are correlated with activity in that network. What's interestingly enough is it's not like area specific. We've had one animal where we had multiple cells firing in PFC. From the same wire that we're recording, where each one of them was sort of tapped into a different network functionally over time. And I'll end the talk on that comment and what we think that might be. But some areas like nucleus accumbens, about 50% of cells were tapped into one of the areas, BLA about 21%. But so something about these networks are capturing cellular firing across time. So they're at least showing dynamics that are relevant. All right, so here's how we're gonna validate this. What you'd like to do in an ideal world is you take a whole new group of animals, C57 animals, bring them in the lab, measure the activity of the network, and then see if it predicts in a whole new group of animals that the model has never seen. So we decided that even if we solved that really hard problem, we would have solved mouse depression, which probably wasn't that useful. And so we decided we're going to do something that's a little more complicated. We were going to use other things that cause vulnerability and see if they made the same network show up. And to be even more complicated, we were going to generate vulnerability by tapping on a different level of analysis on the brain. So we might do something molecular, we might do something cellular, each of those that generate vulnerability, and we're going to see if the exact same network state that shows up. So we're going to take the network that we learned in the C57 animals and see if that exact same network shows up. So we're not going to learn new networks, we're going to take the original ones. So I'm going to speed up a bit in this talk because I'm painting the biology on top of this network structure. All right, so this work was originally done by Rosemary Baggett when she was in Eric Nestler's lab. And this is going to be our molecular experiment. Rosemary basically took the social feed animals, put them through the stress paradigm, took the susceptible animals and the resilient animals, lopped out multiple parts of their brain, and they did RNA sequence, which is just going to tell you about the expression of different genes in different parts of the brain. One of the areas that she was particularly interested in was ventral hippocampus. She looked at differential gene expression patterns and found a gene, sidekick1, which regulated lots of genes that were different in the two groups. So she called this a hub gene. They wanted to ask if this hub gene, overexpression of this hub gene, was sufficient to cause vulnerability or susceptibility. So they created a viral vector, took a new group of animals, upregulated sidekick1 in their ventral hippocampus, and then looked what happened behaviorally. And the animals looked fairly normal on all the behavioral assays, unless you stressed them. If you put the animals through stress, those that had a higher level of sidekick1, like almost all of them showed susceptibility. So they argued in this case that upregulation of Sidekick 1 was generating a latent state in the brain called vulnerability that, if matched with stress, generated susceptibility. So our question was simple. If you took this single brain area and you upregulated this single molecular pathway, would it be sufficient to cause this brain-wide network to show up? All right, so experimentally, all we're going to do is put our animals in this assay. We're going to, after looking at their brain, we're going to give half of them HSV, a control virus, GFP, in ventral hippocampus. The other half are going to get Sidekick 1, and we're just going to ask what happens to this network. This turned out to be a really complicated problem, right? Like HSV expresses in 1 day, it's gone in 8. We were implanting animals with electrodes. We realized that if we did the surgery at the same time, by the time the animals recovered from the surgery, the virus was gone, right? And so we had to figure out a way to basically do viral injections in animals that were already implanted with electrodes in many brain areas. We ultimately figured out how to do this by building cannulas into our recording electrodes. And so again, we're going to take all of our animals, look at their brains beforehand, upregulate this pathway, and then compare what happens when you upregulate the singular molecular pathway or the control virus. And when we do this, we essentially find that we're able to create this brain-wide network state in one of our networks. So we potentiate, in other words, we cause this candidate vulnerability network to show up in one of our networks, network 1. Nothing happens in network 2. So single molecule, single brain area causes this brain-wide network to show up. One of the interesting points that I'll touch on a little bit later is this network, the final sort of last convergent area in this network where the information looks like it's flowing is actually ventral hippocampus, right? All right. The second thing we're going to do is a physiological manipulation. Got into a conversation, Asino Silva, we were at Aptogenetics GRC, and we were talking about interferon alpha. It's an interesting drug. I encountered this on my consults rotation while I was doing psychiatry. And it's a drug that's used to treat hepatitis C. And one of the really nasty side effects of this drug is that it makes people suicidal and have low energy and not eat. And it essentially creates an emotional state that would otherwise be called depression, except we know what's causing it, right? So it's basically one of the exclusion criteria for depression. It can't be depression because we know the drug is causing it. But our hypothesis was, could we essentially generate the same vulnerability brain state, right? About 50%, 60% of people that you saw in this drug have this depression-like emotional state. So what we're going to do is we're going to take a bunch of animals, implant them with electrodes. Half of them are going to get interferon alpha for 5 weeks. The other half are going to get a control virus, and we're going to see what happens to their brain. When you look at these animals behaviorally, they no longer like sugar, they're a little bit socially avoidant, they're a little bit anxious, but we're really interested what happens physiologically to these animals, right? So when we do this, we find that interferon alpha is sufficient to cause the same brain state to show up in their brain, right? So when you put animals through a 3-part assay, the animals treated with interferon alpha have much higher activation of this vulnerability network, and the first network, but not the second one. So the one that didn't work in Sidekick 1 didn't work in interferon alpha. The first one that worked in Sidekick 1 also works in interferon alpha. All right. The next thing we're going to do is take a different level of analysis manipulation. One of the things I encountered repeatedly in my outpatient psychiatry rotation was one of the biggest risk factors for adult mental illness is childhood trauma, right? And there have been multiple people looking at models of childhood trauma, things like you can stress them early in life or separate them from their mom. So we just wanted to ask, if you put mice through the equivalent of childhood trauma, would it cause the same signature to show up in their brain? So now it's a behavioral manipulation. What we're going to do is we're going to take animals when they're born, and we're going to separate them from their moms for the first 20 days of life. We're going to separate them from 3 hours a day for the first 20 days of their life, and then let them grow up normally. When you do this, the animals look reasonably okay on like anxiety assays and social behavioral assays. They're a little bit off in like assays of addiction, but in this case, they look fairly similar. And when you put them through our social defeat stress, what we're going to do in adulthood is, instead of putting them through the standard social defeat stress paradigm, we're not going to house them next to the bully. Normal animals, when you don't house them next to the bully, they don't show any of that stress behavioral problems. When you take the animals that have had childhood trauma and you don't house them next to the bully, they still show all the problems. So behaviorally, they look like they're in a much more vulnerable state. When we look at these animals physiologically, so there's another group of animals that we've now implanted electrodes when they're adults, it basically potentiates network 1. So that vulnerability network is there in their brains before they hit the adult stressor, right? So we've got the molecular manipulation, the physiological, and now the behavioral manipulation, all making this first network show up and all making the second one not show up. the last thing we did was we wanted to search for genetic risk factor because we were being super thorough. Turns out there's no great genetic risk factor for depression except having 2 X chromosomes. So it turns out 66% of the people who have depression are women. And so we just wanted to ask at baseline, is there a difference between how male mice and female mice respond physiologically to this task? And when we do this, it turns out the female animals in pink have activation of the same network in response to this 3-part assay. The CD1s do attack the females. They don't try to mount them. They actually try to bite them through the wall. So same physiological activation. And what was so fascinating about this, these are animals, no childhood trauma, no nothing, just female and male mice. The lowest activation state that we observed in the females was higher than the highest activation state of this network that we observed in the male animals. So it does give some sort of biological argument for why you may see increasing rates of depression in females, because the brain might be more likely to be in that vulnerable state. Okay, so we basically do these 4 independent validations. All of them point to validating at least whatever this signature is as being a predictive physiological marker of vulnerability, right? So we set out to do this, right? We have the disease state, we take antidepressants, we put it in the normal state. We know stress is our manipulation, and we can now segregate these out. And we can now find this thing called vulnerability, right? That we can mark in the brain. And we have signatures of the disease state. We now have signatures of vulnerability. In theory, antidepressants move your brain from here to here. And so the last 4 or 5 slides, what I'm going to spend it on is asking the question, can antidepressants move your brain from here to here, right? If they can, then we should, I gave this talk once and I said we should be putting antidepressants in the water. Someone pointed out they were already there. But the idea there is that we can use antidepressants as a preventative agent. So we're just going to ask physiologically if what antidepressants do to these networks. We have our vulnerability state, which is network 1, our susceptible state, which is network 2 and 3. And we're just going to ask what antidepressants do to these networks. Do they cause their activity to go up or down? We're going to pick 2 antidepressants. We're going to pick ketamine and deep brain stimulation for the sake of the publication. It's because they're really exciting. In reality, it's because we can implement them pretty simply in the lab acutely. Yeah.
I just want to understand the logic here. I mean, digoxin,
Yep.
which is very useful for increasing cardiac output, would not necessarily be a good drug to use in a person before they have
Yes.
congestive heart failure.
Yes, and neither would a pacemaker, right?
Yeah.
Yes, yeah.
So why does this make sense?
Yeah, so on the other hand, statins would be, right? So we're asking if these are the same or different, right? If they are the same, then we're making the argument that we can manipulate beforehand. If they're different, then we have a new biological target to develop those interventions. Yep, all right. So ketamine here, this is just work that was published with Eric and some collaborators, just looking at animals that have gone through social defeat stress, and then they're just asking if ketamine works as an antidepressant. At a dose of 0 milligrams per kilogram, the defeated animals are still socially avoidant. The control animals are not. 2.5, the problems are still there. At a dose of 20 milligrams per kilogram, the animals that have gone through the defeat paradigm now are showing normal behavior. So they're just arguing that 20 milligrams per kilogram of ketamine is antidepressant. So all we're going to do is implant a new group of animals with electrodes. Half of them get ketamine, half of them get saline, and we're going to see what it does to this network. Ketamine causes some real nasty behavior acutely. So we're just going to wait 24 hours, right? So animals get ketamine, we wait 24 hours, and then we just see how their brains activate in these states. When we do this, we find ketamine, unfortunately, does absolutely nothing whatsoever to our vulnerability network at all. Interestingly enough, ketamine hits one of our multivariate encoding disease networks. So ketamine touches one of the disease networks by suppressing it. It does nothing whatsoever to our vulnerability network, all right? The second manipulation we're going to do is deep brain stimulation. This is some of the prior literature looking at the social defeat stress model. They're doing optogenetic stimulation and basically using light to activate cells in a part of the brain. They found that left prefrontal cortex was antidepressant in the social defeat stress model. There was some lateralization here. So we're going to target left prefrontal cortex, and there's some argument that it's infralimbic cortex that mediates this effect. So we're just going to stimulate left infralimbic cortex and see what it does to our networks. We're going to use 2 viruses. One is a stable step function option. In other words, we hit it once with light, and it causes the area to become more active and stay on. That way we don't need to worry about the dynamics that we're using to activate this area. The control experiment, we're just going to take the same virus, turn the genetic sequence around backwards so it doesn't encode anything. So our experimental animals, you can see the GFP, our control animals, nothing happens. When we hit light in infralimbic cortex, we stimulate it about here. You can see that the activity in infralimbic cortex changes. Nothing happens in the control virus case. As an interesting caveat, we've got electrodes everywhere. So it turns out like other areas change acutely also when you stimulate the single brain area. The whole brain looks different when we do this. But you're interested in what happens physiologically. Stimulating prefrontal cortex does absolutely nothing to the vulnerability network. It suppresses the other disease network. So ketamine hits this one. Prefrontal cortex stimulation hits this one, and neither of these do anything to vulnerability whatsoever. So here's a summary of what I've shown you. We basically go through, we learn these three networks in the chronic social defeat stress case, and then we go through this idea of external validation. We show that the sidekick one projects on the vulnerability network, interferon alpha projects on the vulnerability network, early life stress projects on this network, and all of three of these show that the network 2 is not a what we call convergent network. It might encode vulnerability to chronic social defeat stress, but it's not the thing that we would even try to translate across species because it clearly doesn't work across different mouse conditions. All right? And then we do our antidepressant manipulations. None of them touch our vulnerability network, but one hits one disease network, one hits another one. So here's our final model, right? The brain starts off in this what we call a stable, normal state, right? Which is highly tolerant and resistant to major external stressors. And then you can have early life trauma or genetic risk factors, which put your brain into vulnerability. You are totally fine in vulnerability. You can handle life just fine, no behavioral problems, unless you hit a major adult stressor, which then puts your brain from this vulnerability state to a pathology state. Pathology is supported by two pillars, two networks, and all antidepressants have to do is knock down one of the pillars. Your brain tumbles from the disease state back into vulnerability, but it's why disease can reoccur because you don't actually go back down to this state, right? Think about it almost like ice and water and gas vapor, right? Your brain is able to transition from these states, but it doesn't jump back from here to here, all right? So I'll spend the last 2 or 3 slides just talking about where we're going with this and how we're trying to put this all together. We'd like to expand our ability to extract these states out of animals' brains. So we're simultaneously trying to generate convergent models around things like anxiety, right? So what does an anxiety brain state look like? We're working on complex social behaviors, so like social interaction and aggression states. We're trying to decode things like reward anticipation and motivation. And we have an engineering section on the lab that's working on, once we've learned these states, we can actually extract them and quantify them out of the brain in real time. So with about 100 milliseconds delay, we can say how much of this state is present, all of these states are present, and as an animal's running around. So we can have a constant self-report on what's going on in the animal's head. So our strategy was, we started off with this problem, and we think ultimately the way we're going to be able to solve this is by discovering all of these different networks that then map onto the states. And unlike the induced states that we've generated before, we're going to see if these things spontaneously emerge, whether we have genetic models of these diseases. The final piece of work that's being led by Elise Adamson in collaboration with FAN, Elise was recently awarded at HHMI Gilliam, is we're trying to solve that problem that I mentioned about the PFC when we have like multiple cells that we're recording from the same channel that are tapped into different networks. And we think that there might be some important rules about what dictates how a cell becomes encaptured in a network. And so by using microendoscopic calcium imaging, we can ask, does a PFC cell tap into network 1 or network 2 based on where it projects from or where it receives projections from? So we can simultaneously do endoscopic calcium imaging from an area, this is infralimbic cortex, while we're also getting local field potentials and the cellular firing, because we've got both of these techniques integrated together. And so that should allow us to learn the structure function rules, all right? So this obviously happens with a lot of folks from a lot of different disciplines working together. Most of this work was led by Rainbow, who's now at Iowa, David Carlson, who's at Duke, just got a Brain Initiative Theory R1 with our collaborators at other institutions as well. So thanks. Any questions? So in the C57 data set, we've got an AUC of about 0.9, right? So they separate really well in that case of whether an animal will be susceptible or resilient. right? What we're trying to do now, and I like the idea of being able to take an animal and categorize it. What we're really interested in is if you're taking the same animal over time, let's say an anxiety state shows up, how predictive of it in terms of how the animal is feeling acutely and what behavior it chooses, right? Because then we know that these are real encoding strategies in the brain, right? So the reason we really like the real-time extraction is that that allows us to then do real-time closed loop in which we're turning these things on and off and then seeing how an animal chooses difference in behaviors, right? So I think there's a clinical utility to being able to separate animals into groups on whether they're susceptible and resilient. This now allows us to do cool experiments like ask if there are biological factors which mediate that. But the neuroscience case for us is, can we manipulate these things in real time and see how that impacts behavioral choices that animals make? Yeah.
I have a question with respect to the pathology of all of that. So you show that within mice there is a spectrum between those who are vulnerable and those who are not.
Yep.
And presumably evolution has put stress and depressive behavior in there for a reason.
Yep.
So the question is this not just something to be cured and to be removed, but is this also a feature? And should we worry that if we remove this in humans, that we are actually generating an issue that is unforeseen?
Yes, so I agree 100% with you, right? One of the problems with psychiatry, of which there are many, is that one of the criteria for everything we do is behavioral dysfunction, right? And we model and map behavioral dysfunction by how an organism or an individual functions in whatever their immediate environment is, right? And so I've given this talk a ton, and people have asked the question, can we generate new treatments or medicines that we should give babies to prevent things in the first place? And I was like, no, we should let parents off of work so they can take better care of their kids, right? And so part of this is that some of the things we need to tune our environment better, and some things that we call a deficit because of the mismatch between individual environment might actually create advantage in another situation, right? So I'm not making any case about that at all. Right? Yeah.
Kind of alluded to this throughout the talk, but I'm curious to hear more about how you're thinking about the temporal dynamics of all of this, especially when you're trying to extrapolate to humans.
Yes.
How do you know when is the active window of time where you should be looking for these connections? Like, do you think that if you had observed, 1 day later, the mouse that looked resilient would have actually developed the vulnerable state or susceptible state?
Yeah. So there is a time window in which animals are vulnerable to this stress, right? It's somewhere between 8 and 12 weeks, and then they're not, right? This is also the case of many psychiatric diseases for humans in trauma as well. The time window and the temporal dynamics question is a really complicated question, right? Because that could mean seconds or days or weeks, and the brain is actually integrating all of that together, right? So we're trying to solve the question of what is the right time scale and the dynamics in multiple different ways, right? Are there states that are more stable across days, right? And so we're doing longer recordings. We're seeing if these things are present during sleep states, right? And for example, right, are these things, right? So I showed you the average line across groups, and then I said that the individual mice are in the background. Well, these networks are sort of skipping on and off, right? It sort of makes sense if you think about sound, right? You can have information by how loud a sound is repeatedly, right? But if you essentially look, I don't want to clap in the mic, but if you essentially look at the interval right between where I'm clapping, it's silent, right? And so you can think about how, right, the brain or any other system might integrate information across time to create a sound representation that is half of the max and the min. Does that make sense? So we're both trying to solve how long are these present, right? What stage of life are these present? But then also thinking about the seconds time scale. It's the reason why the closed loop stuff is so important, right? Because what happens if we shut down the network at the seconds when it isn't there versus the seconds when it is there? And does that create a latent state, which then makes it harder or not harder to turn it on? I've also made no arguments whatsoever about how these networks interact with each other, right? So I've talked about the things that we call univariate networks. In other words, they show up by themselves and have information. But some of them, it's two things to get, so the information is in two networks together. So we're also giving a lot of thought. I had a great suggestion from one of my colleagues, Elizabeth Hillman, who is doing cool things like turning networks into sound and then listening to them as they interact with each other in an animal's head. so you can start to hear how chords are working together to generate music, right? So we're also thinking about how these networks might show dynamics that are relevant to each other across time. But all of this is sort of new. We just want to prove that they were real to start with. Yeah. All right, thanks.